Hello, here we go. Time for some more practice. Raise your hand if you ever used one of those phones. And if you just raised your hand, let's just like put up two fingers. I'm not going to tell anybody. It'll be our secret. So here we go. I had to make two of these. Okay, so we'll just end this one and start another one a little later. Here we go. Again, take a look, scratch out an answer, think up an answer, whichever, and then click to the next slide. I'll walk you through. Okay, so here we have a very typical story of an enzyme pathway. Very short one. There's only two enzymes here, only two steps, right? But it starts with this precursor, right? Turns it into that intermediate, just using some language here, and eventually ends up with that final product involving the use of two different two different enzymes, which are coded for by two different genes. Okay, so circle one arrow that represents transcription. You might look at this and say, are they kidding? That's too easy. Don't be confused. Lots of things are easy on here. So that's transcription, right? Gene is made of DNA. That's what they're seeing. Do you know? A gene is made of DNA. Do you know that transcription is how DNA gets used to make a pattern for RNA? So you could have circled any one of those. It says circle one. So if you circled two, if you circled both, you would not get a point because, or I'm sorry, you would have to circle both of them right. But if you circled two and one of them was one of these, then you have contradicted yourself. So be careful of that, right? Um, Identify the molecule that would be absent if this enzyme is non-functional. You wouldn't make this chemical. This is really easy, but there's going to be, on this test, there won't be that many of these, but there's going to be some that are really easy like that. Try not to overthink. Here we go. Okay, so they told you that a base pair in the fourth codon has been deleted. Here's what they expect you to know. A deletion of one base pair is going to cause a frame shift. From that point on, every codon is potentially different, and therefore every amino acid, this is usually a bad thing, a big change, right? They said the fourth codon on purpose because they expect you to know that your normal enzyme is a few hundred amino acids long, and so this is going to be a big change, right? You predict very little IAA or probably no production of this, right? And so here you have the idea that if we mess up any part of a pathway because we just messed up that enzyme, because this got mutated, which caused that get mutated, you see the idea here? This is again central dogma stuff, right? DNA to RNA to protein, eventually to some kind of product here, which would cause some kind of trait. They don't get into the trait that. Okay, so your justification is that this mutation is going to result in the translation of a enzyme that doesn't work. Maybe even no translation, right? Or you could go straight to this product, which you could say, well, it's not going to make much of that. You could have said either one of those things to get that 1 point. Okay, another one. Okay, so here, this is a little tricky because they don't tell you this is worth 2 points, all right? But they want you to say two different things here. Explain one feedback mechanism. I would have liked it if they had better said, identify one and explain how it works or something like that, right? Because this point was to identify basically that, oh, this is a negative feedback system. We're going to prevent the making of too much, right? We are going to keep the level of this enzyme kind of steady. This is the negative feedback idea, all right? Now, they tell you we don't want to produce too much of this, but we don't want to limit this. That means this part of it is what must be being inhibited to not make too much of this, okay? Not this part of it. That's why they said without limiting the production of this. Okay, So how could that work? Well, most likely this stuff is either going to inhibit this enzyme, right? Usually allosterically, or maybe it'll even back up and inhibit the transcription of this gene. Could be either way. So the production of the enzyme would be inhibiting the gene to make the RNA to make the enzyme, or the enzyme could already be made, but its activity is inhibited. Different question. Okay, so here we have a codon chart kind of thing, a mutation kind of thing. They describe to you that you have this thing chemical that is a hormone. It promotes stems getting longer, right? Plant growth. This enzyme catalyzes the making of it from a precursor, right? And here we're going to have a mutation in the gene that makes this enzyme, and that mutation results in a short plant. Because without that enzyme, we can't make this hormone that makes it tall. This is all your analysis of the story they're telling you. Okay? So, first, this question is about in the enzyme, they're going to tell you specifically that the wild type allele, and then wild type again means like this is the normal one. In other words, this one works. It has alanine, nonpolar at position 229, but the mutated one codes for a threonine in that same position, and it's polar, not nonpolar. They expect you to know that that kind of change can cause a shape change that can cause that thing not to work, right? This is what they want in the description of the effect of that mutation is they want shape change, but they'll take structure change and they'll even just say it changes the function of the protein, but this is why it changes it. This is definitely better. And if they ask you to explain the effect, you would have had to have said the shape changes because that different amino acid is polar. It's got different properties. It's going to cause different folding, different tertiary folding. You could have gone into that stuff, right? But shape affecting function, that is the key. Okay, so now what's your reason to support how this mutation makes plants short? Again, without that enzyme, we can't make gibberellin, so the mutation decreases, eliminates that. Part B. Just to refresh the memory here, because it's not in front of you, the story they told you said that the enzyme that works has alanine in position 229, and the mutated one has threonine. So they really misworded this, right? The substitution was threonine for alanine, not alanine for threonine. That's a bad mistake on their part, but anyway, just so you're not confused. Okay, so the coding here involves these two amino acids. So first you have to find them on the codon chart. And you see that one is coded for by any codon that's AC anything, and the other is coded for by any codon that's GC anything. So the change was clear. This was a base substitution, right? They asked you to predict the change in the codon sequence, not the DNA sequence, even though the mutation would have happened in DNA, right? They're asking you for that. So they do, however, give you a point if you go back and talk about the DNA change as long as you talk about it correctly. All right, so what you had to have here was a substitution of a G for an A in that first position, and that would have produced the threonine for the alanine. Part C. Okay, so now they tell you that they tell you up here that this is like dominant. How do they tell you that? They tell you you have a pure breeding short one, a pure breeding tall one that makes F1s. They're all tall. When you cross the F1s, you get 3 tall to every 1 short. This is the classic ratios that Mendel got, And this is what you would get if you had a simple dominance kind of situation. So now they tell you one of them's heterozygous. I'm going to use T's, right? One of them's heterozygous for this tallness producing thing, and one of them's homozygous for the wild type, um, uh, or excuse me, or it's got two copies. They're not crossing anything, my fault. Either a plant with that or that is both tall. Well, your answer is, well, if they only have one of these genes, symbolized by the capital T, that makes that enzyme that can make gibberellin, right, then that's all they need. And you could also say, well, they must be able to produce enough gibberellin even with just one of these genes.